I'm Lee Helman, and it's really my pleasure and honor to introduce the honorary speaker of this meeting, Ms. Blackburn. I'm going to try to, she told me to go quick, and it's always more worth listening to what she has to say. She's a fantastic speaker. She received her undergraduate degree in biochemistry at the University of Melbourne, a Ph.D. in Cambridge, working for a fellow named Fred Sanger. I think many of you might be aware of his work. Then she did a postdoc at Yale, and a lot of people don't realize, I confirmed it with Liz because that was mine. It was there that she actually noticed these long 6-mer tandem repeats in an organism, and it fascinated her because she's such an interesting and interested scientist. So then she moved on and got a faculty position at Berkeley, and was really interested in what these were doing because it was clear they had to do with chromosomal integrity over multiple courses of duplication. And so then working, and so she had to figure out how did these repeats get replenished. And so working with Carol Reeder, they identified an enzyme called telomerase, which in fact depleted the chromosomes so they didn't disappear over a few cycles of replication. They received the Nobel Prize for Physiology and Medicine for this work, along with Jack Szostak, I hope I said his name right, in 2009. And she has continued to work on this, and suffice it to say that telomeres and telomerase affect almost all of biology, including cancer, aging, and many other effects on normal biology. So she's been asked to be on so many committees, and she supports so many groups. I just one thing that really struck me, I'm a big fan of medical history, and I noticed that during Bush's administration in 2002, she was asked to serve on the President's Council of Bioethics. And that was a time when there was a real issue about stem cell science. And Liz was a stalwart in saying, we need to do this. So her integrity and commitment to doing good science was shown there. And it's been my pleasure to work with her on the Scientific Advisory Committee of Stand Up to Cancer, where she continues to show that fervor for doing good science and how important doing good science is. So we are really lucky to have, one of the pillars of our great scientists that came from Australia and moved to London and now is working here in the Bay Area. She's currently, I can't even remember your position. I wrote it down, but I know what she is. She's the Morris Herzstein Professor of Biology and Physiology here at USC. And as I said, she serves on so many committees, I don't want to go through them. But she's a real asset to our scientific community here. So thank you, Liz, and the podium.
Thank you, Lee, for this much too gracious introduction. I'm just so pleased to be able to join you here, everyone here at UCSF, and where I too, like a number of members of this consortium, I've had the privilege of many productive and collegial collaborations over the course of my career. And so I want to, in turn, say a few things about PNOC. UCSF, after all, it's a center of gravity for innovation in things including oncology. And so it seems sort of natural that the Pacific Pediatric Neuro-Oncology Consortium, which I will refer to as PNOC from now on, that it can call this institution home. And as of course you know, it was co-founded by my UCSF colleagues, Sabine Mueller and Michael Prados. And I'm just really impressed by how PNOC has established itself as a locus for cutting-edge clinical research and a real resource to the pediatric uro-oncology community around the world. Now, it's the 10th anniversary, so this is no small matter. Because when an academic research consortium celebrates something as long as a 10th anniversary, this is really saying something because, as we all know, big, complex, multinational academic research collaborations, they're complicated entities to establish and to manage and to grow and to sustain. And some are more productive than others. And it looks like, on all those criteria, PNOC really does earn high marks. Because when you think about it, making real progress in a challenge as tough as pediatric cancers, and that's what the consortium is doing, well, this means you have to creatively disrupt the status quo in some ways that are strategic and unique. So what ways? Well, collaborativeness, obviously. PNOC is, I think, inspiringly collaborative, harnessing the power of partnerships to advance discoveries, breaking down silos that inhibit communication and progress, making cooperation something that's incentivized among clinician scientists and researchers, and partnering, being productive about it. PNOC does this with the Children's Brain Tumor Network, for example, led by Adam Resnick, applying the latest findings in brain tumor biology, and then designing what we hope will be ever smarter and ever more high-impact clinical trials, and advancing with real speed, urgency. I mean, this is demanded by pediatric cancers. There's such an unmet need. It has to be advanced as fast as possible. And finally, being global in reach. And as you have heard, PNOC is indeed very global. I felt very privileged this morning listening to the panel on so many challenges and how you reach people who need to be reached to treat pediatric cancers, particularly brain cancers, this morning. And this is just, encouraging as well as daunting to hear the ways that this is being addressed. So I think I'm really hopeful this really will be making a big difference. And I also want to shout out the PNOC Foundation, because not only have they raised much needed financial support, but they're also raising awareness. Again, as you heard this morning in the panel, so important. And so I want to congratulate particularly Bruce and Alan Campbell, because it was they who were inspired to establish the foundation for PNOC. When their son George, who now happily is well, was diagnosed with an anaplastic ependymoma. So I want to wish the foundation and its board leadership and its professional staff all the best as they're launching a Lose No Child campaign, and raising more philanthropic support for pediatric brain cancer research. This is exciting, and this is really boding well for the future of the consortium. And Bruce and Alan, I just want to personally wish your son George great success and happiness as he embarks on a new phase in life. And my understanding is that, in fact, he's just started college, and this is a real milestone for parents under any circumstances, especially meaningful in your case, and really movingly, I think, emblematic of the progress and promise of pediatric cancer research. So when Sang Pobliti of PNOC, when she asked me if I'd be willing to participate at this important gathering, I said yes. And then after I accepted this kind of invitation, then the organizers said, would I also talk a bit about my the relationship between my work and this is on, as Lee's pointed out, on the structure and function of telomeres, and the relationship of that work with pediatric uro-oncology. And actually, it's a particularly, for me, very real relationship. So I'm going to switch gears now, and I'm going to have a laser-like focus on this aspect of pediatric brain tumors, pediatric uro-oncology. And it's going to be, about telomeres. But you'll see that this is actually an important aspect, it turned out, in the biology of these various kinds of tumors. And you'll see that it actually is important in stacking the deck, pointing, and finally, as I'll point out, pointing the way to more avenues for hope in improving treatments. So let me just get on with now diving in, as I said, with a pretty laser-like focus, but ending up from going from pretty basic science, talking about, well, what is the current picture of telomeres in pediatric neuro-oncology, something about causality. I won't spend a whole lot of time on that, but it'll emerge as I talk about this, and then implications for treatment. So let's just dive right in. Very quick tour through telomere basic science, beginning with an organism in pond scum in which this basic research began. From such humble beginnings, I would never have guessed I would be so fascinated by how this has led so directly into pediatric neuro-oncology. So here's a ciliated protozoan. There's a cell, has a nucleus, and it has, in fact, lots and lots of chromosomes, which enable me to molecularly look at the very end of the chromosomes. They have these repeated sequences at the ends. In this case, G4TT repeats. In humans, same idea. It's just one of the G's is replaced by an A. So see, that's the telomere. And a lot is known about how important these things are at the ends of chromosomes. They basically protect the ends of chromosomes. And not only is there DNA, the DNA has a whole sheath of very important, critical protective proteins. I'm not going to go into names. We're going to stay pretty high level for most of this talk. But the point is that if telomeres get too short, that's a real problem. And what happens is the shorter telomere looks to the cell like a broken chromosome, and it says, oh, danger, DNA damage signaling. It becomes very persistent signaling. Long story short, the signaling tells the cells to stop multiplying in a variety of different ways. It can also lead to genomic instability in certain genetic settings in the cell. And that's actually, you can make telomeres non-functional in different ways, but this physiologically is just the most important and common way that telomeres get too short. They get too short through a variety of reasons, mostly known as the incomplete DNA replication at the very ends of linear chromosomes. There are actually other ways. There's DNA damage that occurs, replication forks break it as they go through telomeres. There are other ways that telomeres can get more abruptly short, but basically they have real propensity to get shorter. So the problem was how can they get elongated, and that's what Carol and I discovered way back in the 80s. And we called the enzyme telomerase, and it actually makes telomeres longer. And the mechanism, end of a chromosome shown in black, is a fascinating one. There's an RNA built into the complex of lots of proteins. I'm just showing a simple version here. And what happens is that the RNA has a little template, and then DNA synthesis elongates the chromosome end. So the whole picture is that we've got this mechanism, and you've got, we found it in tetrahymena first, it's all throughout most eukaryotes, a few exceptions, and it's a reverse transcription mechanism, which is kind of a curious one, and it is templated by this RNA component. So if telomeres get shorter, then telomerase is attracted to them, and it adds repeats to the end, as I just indicated, and then the other strand is filled in by discontinuous synthesis, using the DNA replication machinery here, and the gaps are partly sealed up, and then you get a long enough telomere which is capped. Right? And so, very nice, did this matter? So in tetrahymena we could do the simple, very surgical genetics of in a cell which actually is effectively immortal, just keeps on growing and growing. Telomeres are maintained, and if you surgically kill the enzyme's activity, then the chromosomes got shorter and shorter. So this was the essence of it. And the cells died. Right? So, okay, so there we were in the ciliated protozoan system. But what about in humans? Well, yes, the enzyme exists, but it's a much more complicated regulatory story. every mechanism of regulation you can think of gets called into play both in normal cells during development and various sorts of environmental and physiological prompts, and as you'll see in cancers as well. So it's highly, highly regulated activity, really regulated. Just to remind you of a couple names, particularly the core protein TOT, because that gene will come up as I go on a bit. So the bottom line, without enough telomerase, most normal cells have this propensity. Eventually it leads often to senescence, other ways of cells not dividing, cells malfunction, there's a lot of signaling changes in how cells function. And, okay, so just to reiterate, we have this situation. There's a lot of ways cells don't function well. People have looked at these, and these can explain some of the physiological effects of when cells lose telomere maintenance. Particularly senescence is interesting because it sets up all sorts of programs that can then affect other cells by secretion of pro-inflammatory and other factors. Okay, so that's some telomere basic science. Okay, let's home in on pediatric neuro-oncology now. And thinking about cancers, this telomere maintenance story is clearly a two-edged sword. Okay, so I've just told you, about replenishment by telomerase. This allows telomeres to be protected. This is good news in at least the normal healthy cells of the body, and particularly good in normal stem cells and highly proliferating normal cells. They have telomerase, sustaining their telomeres. Okay, but of course the converse is really bad news, because now these telomeres being maintained gives cancer cells permission to keep on multiplying. That is the key downside of this in real life, in real humans. So what we see is in a non-cancerous cell, as I've just said, telomerase pretty much often repressed. Cell divisions eventually will stop. But as they progress into cancerous states, and of course mutations accumulate, and I'm not going to be talking about the critically important cancer-driving mutations that are going on here. As I said, we're going to be laser-like focused on the telomere maintenance aspect. But of course, cancers have mutations, including pediatric brain cancers. And so, in humans overall, telomerase, strong upregulation, is a really popular way that human tumors keep their cells able to multiply, because the telomeres are not running down, and so they have permission to keep multiplying. Okay, so that's a really popular mechanism throughout the whole spectrum of cancers, throughout humans, throughout human lifespans. Now, this is really something that stacks the deck clinically. And I'm just going to give simple examples as I go through in the setting of pediatric brain tumors. And so this is a paper back from 2006, but it really makes the point that in pediatric intracranial ependymomas, if you look at Kaplan-Meier survival curves, this is shown in a pretty small cohort. It's held up basically if telomerase has been upregulated by whatever means, we'll hear some of them in a moment, then those patients have much worse prognosis, as you see in this progression-free survival or overall survival Kaplan-Meier indications. And then you can even divide it up, as they did in this small study, with different categories. What about grade 2 versus grade 3 tumors? You can just see there's a stark difference between the ones that had activated telomerase versus the ones that hadn't, and even gross total versus just subtotal resection. You can see having telomerase activated versus not was a real significant stacker of the decks. So, and again, I'm not talking about all the other important mutations here. We're just looking at this single parameter here. Okay, here's another study in high-risk neuroblastoma. This is from a more recent, I think this is a 2020 paper, and first of all, you can look again at the overall survival, and you can see if TERT, that telomerase component, was expressed high versus low, you can see again a real clear difference. And by the way, this held up even with this common occurrence in neuroblastomas, which is MIG-N amplification, still in that subset of patients in which MIG-N was amplified, still you could see that big difference. So having telomerase was really a stark difference here. Okay, but that's not all. So years and years ago, Vicky Lundblad, working with yeast, found that there was this interesting thing that happened to yeast cells. So we're going to have a little basic science here, which will take us right again into pediatric brain tumors. So she found this interesting thing that if you don't have telomerase, there's other ways that cells can call in to maintain their telomeres. Now, the general class is called ALT, for alternative telomere lengthening. The alternative is to telomerase mechanism for maintaining telomeres. And that turns out in childhood neural cancers to play an outsized, disproportionate role, right? It's really an interesting thing. And we were looking at it, as this sort of almost curiosity in yeast. And so, as I said, telomerase-independent pathways, and we found it in budding yeast. Now, there's two forms, and I'm just going to go through them, so bear with me, because they actually do really inform us about what's going on in childhood cancers. And so I'm going to take you through them. ALT2 is the closest kind of ALT mechanism in yeast that's closest to what goes on very commonly, or is at least best understood, let's say, in pediatric neuro-oncology, and then ALT1. So let's what is ALT2? Well, it's basically, as I said, telomerase independent, and yeast cells go into a sort of epigenetic state when they've lost their telomerase, and some of them can flip into this state where now they rely on telomere maintenance, and it's a recombination dependent. Reagenetics shows this, and it's called break-induced replication, and there's the pathway to a telomere that's too short, right? So now it's sort of uncapped, and it resembles DNA break. And as happens in internal regions in chromosomes when there's a break, they can have an option of homologous recombination. And so it invades a longer telomere, which has got the same sequences it can invade, and then by DNA repair synthesis, just like in break-induced recombination in other parts of the chromosomes, it copies a telomere, and especially if it's a longer telomere or it's misaligned, so it lines up differently from the short telomere along the array of repeats, and it can copy all the way to the end, and you get a long telomeres. And they can be very variable in length because these crossovers can take place in different parts. That's the hallmark of ALT that's very strikingly obvious in pediatric cancers. The telomeres are generally long but highly heterogeneous in length. And by the way, we looked at these yeast cells, and on the face of them, they look like they're growing really happily. They're growing well, right? But we did transcriptional profiling, and guess what showed up? A sustained environmental stress response, which didn't go away, even though the cells were growing really well. So these cells are growing well, but they're kind of hurting inside. They're actually under continuous stress, this population of cells. Kind of interesting. Okay, so now, as I said, there's two forms. So let me just briefly outline ALT1. Now this is a little more complex, and it might look yeast specific, but I think it's probably pretty relevant to aspects of ALT in pediatric cancers. So ALT1 relies on a particular architecture of the sub-telomeric region in budding yeast where we discovered this. So you have these long elements, the green DNA, the 5-6 kb long stretch of DNA, and it's called Y prime element, doesn't matter. They only found subtelomerically. And they're bounded on both sides by a little short tract of telomeric repeat. You can see the red there, and the telomere on the other side of it, okay? So lots of the telomeres have this. And I'll just draw it for simplicity without the double strand, just keep it simple, okay? So now we inactivated telomerase, as I pointed out, and a subset of the cells do this. telomeres shorten, as I said they would. Now they become subject to invasive recombination events. And so if another telomere with Y' element lines up, you can get a crossover event between that terminal repeat and first of all that little internal tract, and then now you've got a couple of Y' here. And so now they can misalign again, and now you can get an unequal crossover, and you can get really long arrays of the Y'. That's what happens really frequently in a subset of the telomeric repeats. And you say, well, why does this help? Because it actually is not making the terminal tract of repeats any longer. But it does something else. It sets up a big heterochromatin domain. That's what happens in cells when you have big repeats. And this was shown beautifully in Drosophila first, that's enough to set up heterochromatin. In fact, Drosophila just relies on this kind of heterochromatization for its telomeres. Doesn't mess with this stupid telomerase stuff. In fact, it doesn't even have it, right? So no repeats of the classic kind in Drosophila. It just heterochromatizes telomeres. Why this is important is that, as you know, cancer cells, or as you might prefer, cancer cells are going to use every way they can to protect telomeres. And so this, in fact, is a real way of thinking about this. So I'm just going to go through the yeast part just again. So we have wild-type telomerase, telomerase plus, you have telomerase is the way telomeres are maintained, cells are growing just fine. Long-term, the population growth continues fine. Take it away, the cells will run the telomeres down, they'll go into senescence, and so they won't have a long-term population growth. But actually at very decent frequencies, we estimated 1 in a few hundred, 1 in 1,000 or so, you get either the ORD1 or the ORD2 telomeres coming out. They both grow, the populations grow well. They're stressed, but they're growing well. The genetic requirements, they're distinct, so they both require that recombination protein RAD52, and then they have some distinct genetic requirements. So we understand this quite well, and that's quite helpful for when we look at things going on in a much more complex way in human cells. So just to sit back for a minute, we have telomeres often shortening. Telomerase can elongate telomeres, good, but in humans, of course, the situation is in many normal cells, many of which, normal cells will be the ones that eventually, some of which will accumulate mutations and cause cancers. Telomerase is limiting. So telomeres can get too short. And the point I want to make here is you don't see all going on in human cells when they're normal cells. So right away, there's a cancer cell difference that you'll see. So what is striking about pediatric cancers is that even more frequently than in the general population of cancers overall, ALT is the way that the cancers acquire telomere maintenance. And that 10 to 15%, which is over the entire spectrum of all cancers, a lot of that is actually accounted for by things like brain tumors and pediatric brain tumors. Now, we talked about recombination, and I walked us through that. But in fact, heterochromatin, as I'll point out later, may be another really interesting opportunity for attacking this aspect of what's making cancer cells keep able to grow. But mechanisms are still somewhat mysterious, I would say, but we've got some real inroads, and I'll show you. Okay, so let's just look at a few examples, a bit more specifically, of different kinds of pediatric brain and neural tumors. So quite strikingly, you see that there are 3 recurrent genetic aberrations that you find in high-risk neuroblastoma, high-risk neuroblastoma that is, and they're weekend amplification and turn rearrangements. Both of those have the result of activating telomerase activity. Now, there's another distinct class, ATRX mutations. So what is ATRX? Long story short, it basically is related to heterochromatin properties and is required for certain heterochromatin properties of cells. We don't need the details. That's just such a recurrent mutation. And the important point is that they're mutually exclusive. The cell doesn't really care. Okay, telomerase or alt, I don't care. I just want to maintain my telomeres, right? That's the thing that's being selected for. And so the mutual exclusion is either one class red or the other class purple for alt. And 20 to 25% of mutations, HRX mutations, I think is a rough number. So let's look at metastatic medulloblastomas. Similar sort of story. You get either, in this case, promoter mutations or patterns of methylation. Again, upregulating telomerase or ATRX mutations. Again, same kind of thing, either one or the other. And pretty high frequencies. You can get promoter methylation and mutation rearrangements simultaneously, so those 20 and 60% don't have to add up, right? But you can see both in some cells. Or you get ATRX mutations and something like 30%. So these are major recurrent kinds of changes. Pediatric high-grade gliomas, similar story, where again you get one or the other sets of recurrent mutations. And some numbers, you can do it through promoter mutation or methylation, both of those upregulated telomerase, ATRX loss in about 30%, and in about 40%, some of which also have ATRX loss. Histone H3.3, specifically H3, F3A, okay? And a subset of those can have ATRX loss as well. So the whole point is the cells are selecting for telomere maintenance. Now this is really pointing us, histones, right? We're talking right at the center of chromatin here. So now I'm going to suddenly switch, look, I made it dark so we can all, so I'm going to suddenly switch to much more the present. And this was a few years ago, but we, we did these studies because Tracy Chow in the lab was really interested in, well, what is this about the heterochromatization of telomeres? It had never really been something that had been dived into all that much in at least human cells. And so we had some great collaborators listed here at UCSF. And now, if we look at telomeric chromatin, instead of that slightly simpler picture I showed you where it's just covered, the telomeric repeat tract is just covered with the so-called sheltering proteins, these specialized proteins that bind sequence-specific on the telomeric repeats. Actually, a dirty little secret is that the telomeric tracts are also covered with nucleosomes. We don't quite know if they can, how they're arranged, but they're both there on telomeric tracts. They coexist in some way. So we have chromatin on telomeres, and that's what it looks like. And so Tracy said, well, let's locally manipulate telomeric chromatin and make it more heterochromatic. So heterochromatin and telomeres. As I mentioned, Drosophila actually that's the natural way it maintains its telomeres. It doesn't bother with telomerase or telomeric repeats. It just sets up a heterochromatic domain at the ends of its chromosomes. And in genetic mutation situations, you can see this going on in another yeast, fission yeast, even in certain mouse telomere situations, you can see this. Heterochromatin histone marks, notably the H3K9-methyl-3 mark, that's prominent there, and that's strongly related to this protein called heterochromatin protein 1, HP1, and that produces high affinity binding, and then that allows an extension of the heterochromatin domain to march along where HP1 has been bound. And you get this spreading effect here. Okay, so heterochromatin is certainly not a foreign idea at telomeres in terms of functionality. And humans, it's a bit more kind of, you can find the histone marks at telomeres, but HP1 is pretty infrequently, it's not that obviously present on the telomeric repeats at all. And also you can find, some of the euchromatin marks at telomeres. So it was always a bit, what's the problem? We don't quite understand this. It was a bit of an in-between world, actually. But because there is at least some HP1 at telomeres, we said, well, okay, it's not an unnatural thing to try and really put HP1 much more in telomeres and ask what happened. So what if we alter the human telomere chromatin by dragging more HP1 there. So that's exactly what Tracy did. This is the idea, you make it more heterochromatic and say what happens. So that's exactly what Tracy did. She engineered this controlled system to alter the chromatin states, and she made use of the fact that some of the sheltering proteins that bind along the telomeric repeats bind telomeres specifically. And so she could fuse HP1 to one of them. It happened to be one called TRF1. And so she made a fusion, and long story short, she could actually drag it onto the telomeres, and you could find that the telomeres did indeed get enriched at HP1 alpha. So she checked her system. Yes, she did, it enriched it. She I'm not going to walk you through all the data. It's all published, but you could do exactly what that diagram says. You could make telomeres much more heterochromatic, enriching HP1. You could see the marks appropriately, including H3, K9 trimethyl, and other marks as well went up on the telomeric repeats. You didn't mess the telomeres up, actually. Critical sheltering protein TRF2 was fine. It wasn't kicked off or anything. There was plenty there, and there was no damage. So we definitely protected the telomeres. And then we said, well, actually, let's ask 3 questions about the function. Did we protect the telomeres? So we asked that in 3 different ways. It did indeed control telomerase extension, and that's exactly what a well-regulated telomere protein complex does. It regulates and controls telomerase extension, and in fact, this over-controlled it a bit. It protected telomeres from being damaged, and it actually impacted the structure. But the important point was that it did protect telomeres from being damaged. And so I will later, at the very end of the talk, come to, okay, this is an opportunity for a therapeutic target, and I'll show you a little progress we made on that. So now we really think that we have to include heterochromatin in our thinking about how ALT is working, because ALT is a fairly broad brush term, and we've got recombination mechanisms, but others too. So the consequences can be outlined like this, and particularly in neuroblastoma. And as you're all aware, there's the forms of neuroblastoma that spontaneously regress, spontaneously. And what is so striking about these ones is that they have not acquired any telomerase or ALT mechanism for telomere maintenance through, for example, these recurrent mutations and the natural shortening processes that take place. They take place, the telomeres get exhausted, the reserve of repeats runs down, and they spontaneously regress. So in other words, this is saying if you can stop telomere maintenance, neuroblastomas will regress, and we'll talk about the specifics of that. Okay, so now if you there's a somewhat distinction clinically and in the course of the diseases, which are telomerase positive versus the ALT positive versions, and that's captured very simply in these graphs here, where somewhat earlier, perhaps, are the telomerase positive. These are stage 4, they keep growing out, versus the ALT positive, somewhat slower, somewhat more prominent, perhaps in younger adults, but still this sort of real growth. And now in high-risk neuroblastomas of the kinds I just talked about, I just had earlier showed you those curves, the Kaplan-Meier survival for just thinking about telomerase. Well, what about ALT? And if again you look at overall survival of the ones that acquired ALT or acquired telomerase. But if you look at those patients who had an event in that graph on the bottom left, and you now look at the ones who had an event, what about their survival? And again, ALT and telomere maintenance by telomerase were really bad news compared with not. So this is really quite striking. This is a real stacker of the decks, whether you've activated or not telomerase or ALT. Okay, and so this big difference persists even in the patients who did have a recurrence event. So what might we say about causality here? I'll just go very briefly through this. It obviously falls into a couple of different categories: genetic predisposition or somatic cell mutations, the mutations occurring as the cancer develops. So causality, just to briefly comment, genetic predisposition. Now, people have done big GWAS studies, not only for overall human cancers and subtypes of adult, but also in pediatric cancers. And it's interesting because there's a real, but it's not huge, but it's statistically discernible risk of childhood brain cancers occurring if you have the whole bunch of genetic, small variants that make your telomeres just a bit longer. They're just the normal population ones, and you can see a shift in the probability. But this is not a huge actionable effect at all. It's just saying, yes, because you're looking at genetic and Mendelian randomization things, you can say, yes, there's some causality there. It's this telomere maintenance. Telomeres are a bit longer. That's probably giving the cells a longer runway to accumulate mutations before the telomeres run down, and so they can have a higher chance of forming cancers, including pediatric brain tumors. So it sort of highlights, yes, there's an important role of telomeres, but it doesn't change things in terms of, these are not big odds ratio changes in this population studies. So somatic cell mutations, I've really referred to them. In fact, we can see changes happening in the genome which actually upregulate telomerase or ALT, often mutually exclusively. They don't have to be mutually exclusive, but that's by far the more common situation, as I understand it. And again, each one is associated with not only the poor prognosis that I mentioned, but also metastasis as well. Okay, so what about implications of this for treatment options? Because we know there's still a long way to go in optimizing treatments for these kinds of tumors. Obvious targets, telomerase and ALT. This is a real brief summary, and I'm sure many of you in the room are much more up to date on this, but it's a brief summary of these that I just got from a fairly recent review. So, forgive me, but it just gives you the flavor. So you can say, well, it's pretty obvious. Shouldn't we just inhibit telomerase? Sounds like a good idea, excepting, and there are some really good specific inhibitors of telomerase in various ways, but it's not such a great idea because basically the rest of your body notices if something comes in and inhibits telomerase, particularly the hemopoietic system. And so, you get side effects. Maybe if there could be tremendously well-targeted delivery of these drugs, yeah, this might make very good sense. And those cells that have used the telomerase activation for upregulation. Although they will have ALT, potentially up their sleeves too, as resistance too. But there's various small molecules. You can attack the telomerase inhibition from the end of the chromosome aspect by stabilizing something called G quadruplexes, which is a secondary structure that DNA can form, and telomeric DNA forms it, and it kind of curls up into this very blocky little structure that blocks telomerase activity. Or you can use a nucleoside analog that telomerase particularly likes, but your DNA replication machinery doesn't like so much, and it's this one's a 6-thio 20-deoxyguanosine, 6-thio-DG, and some hopeful studies in model systems. So there could be things that work there. But again, there's real downsides and potential side effects here, as has been seen in trials of some of these. So what about ALT? Well, interestingly, in those ATRX fusion protein mutation in neuroblastomas, there has been some actual, progress made in clinical testing, and there is a trial. And again, I'm talking to people who know this already, but this perhaps could be pushed forward. And then again, other things that do inhibit ALT. But sometimes you don't know the mechanism. That's not going to be so easy for regulatory purposes. ATM inhibition, that's actually an interesting thing because ATM is required for some of the DNA damage repair processes. And curiously, that the observation was that when you inhibited ATM, then at least looking at neuroblastoma cells, which were old plus, those that were resistant to a couple of chemotherapeutics that I listed up here, they, you could overcome this resistance. So there's some interesting, possibilities here. PARP inhibitors, again, a lot of good clinical possibilities there. Again, particularly in combination, and some, at least in vitro models for the ATRX mutant class of old cells, neuroblastomas, possibly, some potentials there. So, epigenetic. So I've been sort of leading you to think about heterochromatin again, because I think that that's something that we can think about through the lens of telomeres. Now, we normally think of epigenetic deregulators as doing what, of course, we know they do, which is to have these, big effects on transcriptional profiles, and so you end up with different programs going on, some of which can then affect the progression of cancers. So there's a lot of things that epigenetic deregulators can do. But we, and particularly Tracy, thought, well, why don't we focus in on the telomere damage part? We know cancer cells rely on telomere maintenance pretty much for maintaining the cancer. And so we were looking at this situation, as I said, where we made the telomeres very heterochromatic and they were very protected. Okay? So what if we could attack that aspect of it, do the exact converse? And so Tracy and colleagues here at UCSF designed a high throughput screen. And basically, it was to look in a kind of agnostic way at a couple thousand different molecules. I'll show the libraries in a moment. And basically, saying, let's actually focus in on telomere damage, short-term damage, because that's the way you can stop cells in their tracks if you really damage the telomeres in a short term, not just let them run down over time or something, but we want damage right away. So in the compound, she added them to a couple of different cell lines. This was high throughput screens. One of them was a brain cancer cell line, and then we used high quality image analysis after staining for DNA damage foci, telomeres, and the DNA damage foci that were on telomeres, right? So specific telomere damage, we call them TIFs for short, and then we look for which compounds show, in just a couple of days, this dramatic effect on increasing damage foci. And so she and Michelle Arkin at UCSF's drug screening facility looked through, and this was the library. It was a couple of thousand compounds. One of them was an epigenetic library of about 100 or so compounds, and there was a couple of thousand other ones, which included a lot of already approved drugs. And to bring it to a short conclusion, look what popped up. We didn't ask for this. The top hits, if you want to do short-term damage to telomeres, looks like epigenetics targeting drugs are good ones. And these are ones that are no strangers, so there was nothing novel about them. So this is interesting because I think this might be raising another viewpoint on why epigenetic drugs, for which there's often a lot of really good rationales, but based on transcriptional control, might be quite interesting in this regard. And so that's where we are. Let me just summarize that. I walked you through a bit of basic science and how that informed, I think, quite surprisingly, childhood neuro-oncology in a way that really wasn't expected, why there would be such a prominent role and impact of altered telomere maintenance for outcomes in pediatric neuro-oncology. It's really quite striking among the whole spectrum of cancers how clear, as you see, these differences were. And we talked a little bit about causality, and it's all clearly saying, it's the telomere maintenance that's the important thing here. The nature of the somatic mutations, even the statistical difference for the genetic predisposition. It's all saying it's the telomere maintenance itself. So it seemed sort of axiomatic to me, we should take advantage of that and try to see if that can be used in, we hope, a way to add more hope to treatment for this very important set of childhood cancers. And so that's all I have to say, and I just want to add my congratulations to the consortium and to the foundation. Thank you.
Are there any questions from the audience? Paul?
That was a lovely talk, and I think a really great synthesis of a topic that's pretty confusing to a lot of us on the pediatric brain tumor side, so I really appreciate that. You pointed out in the neuroblastoma data that the patients that didn't have ALT and didn't have telomerase upregulated did better. How are those tumors maintaining their telomeres? Do we have any insights to that? Is that different by different cancer?
Some neuroblastomas can kind of, this is why I said mysterious mechanisms. They may be kicking in these heterochromatic mechanisms that are not obvious by the ATRX, and I didn't mention, DAXX, the other class of mutations that you associate with all pretty much the same kind of thing going on. But they have, I think, up their sleeves this heterochromatic mechanism, which is much less, it's much more, sort of vague about what's really going on. But as I said, it's so clear in Drosophila that that's the only way they do it, right? That we know that that's a viable mechanism for maintaining, at least some measure of telomere stability. So yes, I'm sure there are complications. I haven't mentioned the other driver mutation settings in which all of this plays out, which I imagine will also play out in ways that might affect, being able to maintain telomeres in certain ways. So I really was sort of pretty high level, broad brush ATX telomerase positive, alt telomerase positive, but cancer cells, as we know, are wily beasts and they'll use whatever they can. So I suspect heterochromia. Hit them with an epigenetic agent, see if it kills them. That would be my strong thought about that.
So in pediatric cancers, in contrast to adults, you're paying attention to natural mechanisms, evolutionary pressure, as opposed to onslaughts of environment, and potentially the role of cancer in constraint to evolution. So when you think about telomeres, do you think of them as sort of a core solution to the end replication problem, or is it really that it's a control framework against cancer as an evolutionary framework, right? Do we need telomeres as a way to keep cancer at bay? And related to that,
yeah, the answer is yes to both.
The second question is, there are differences in mechanism of control of cancer between, let's say, small organisms and very large organisms that have had to contend with large-scale proliferation of cells between elephants and
exactly. Right. So that was what's so interesting. I mean, the small organisms, including mice, I always say, they give us the menu of molecular and cellular possibilities, right? There's a menu. And now we have these long-lived organisms, and they can choose aspects of the menu that will work in terms of whatever, led one to be long-lived. So the whole telomere thing is, I showed that by this sort of yes, no, good, bad, double-edged sword analogy, yeah, they're wonderful for normal cells, but they give them a runway from which to do mutations. So now you could say, well, let's fix mutation rates. And I'm fascinated because, for example, Leona Sampson's work at MIT, she's shown over years, she's studied DNA repair mechanisms of the kind relevant for mutations that accumulate, including others, and if you over-regulate DNA repair, the cells go, there's too much cell death, and for example, photoreceptors in the eye, no, there's too much cell death. So what do you want to take? Blindness? Cancer? So in other words, it seems like we're on this real kind of balance. And genetically speaking, my associate many years at my lab at UCSF, Jolene, collaborated with people in Europe, and it was only a European cohort, and they said, we know that there's a spectrum of genes that in common population situations make telomeres somewhat longer or somewhat shorter. There's huge amounts of data for dailies, so adding up all of their disability as well as years lost to diseases in these databases. Let's ask if longer telomeres are better or worse, and for what kinds of causes. Lo and behold, the longer ones are better for cardiovascular, for avoiding dementias. They probably interact with lots of other factors. They're also worse for cancers. The dailies in that European population, looking at this elderly population as it ages, actually, it's about a wash. So we're right there. Now, I always like to point out that people say, well, what about things that make your telomeres longer? And I say, at least exercise never gave anybody cancer.